Okay, so this video is to train you on how to identify an unknown staph species and really familiarize you with the biochemical tests that would be common for a staph, a suspected staph infection. So I'm going to walk you through unknown 1 and show you how to read the tests, and then I have 2 practice PowerPoints, and then you can check your answers in the module. So we're going to look at some of the characteristics of staph, how the clinical specimens are collected, and then the different tests, the biochemical and the physical tests, to identify the species of staph. So on your own, you can click on these links. When you open up the PowerPoint on your own, you can look at these videos of how to collect the samples, but staph is pretty common in the nose. And so, of course, if you had a staph infection somewhere else, you could swab that. But if you swabbed your own nose right now, you would find some staph. So that's what we would have done in the lab. And then what we're looking for, staph has these characteristics. So staph would be gram-positive. They are cocci, and they are in clusters. And so we would be looking for those things. Also, staph is catalase positive. I'll be explaining what that is. Halotolerant, you'll remember that means it can grow with high salt concentration. And then it is either a beta or a gamma hemolytic. So that one we'll need to address as well. So I've circled all of these things we can determine with a gram stain and then look at it under the microscope. I am going to explain the catalase test. Halotolerant, you can see if it grows either in a salt broth or a salt agar. We're going to use a salt agar. And then beta or gamma hemolytic, you're going to use something called blood agar. So the gram stain you're familiar with, the growth in salt you're familiar with, but the catalase test is new and the blood agar is going to be new. But these would be things that would identify something as staph. So when I remove those, it shows you the 4 things we need to do to test for staph. And this little recording sheet, or you can just write down the results as we go through, and then there's going to be a chart you can look at the end. It's a very similar concept to how we did the Enterobacteriaceae unknowns that you wrote your write-up on. You're not going to write a formal write-up on this, but we're going to practice being able to read the results and then figure out a species for staph, and then in a different module for strep. So the first test, or the first media that we will use, is called a Mannitol Salt Agar. This is really 2 tests in one, because mannitol, you might remember that phenol red mannitol broth that we used earlier in the Enterobacteria unknowns. And this is the same thing, except it's an agar. If the specimen can use the phenol red, or excuse me, if it can use the mannitol, if it can ferment the mannitol, then it will make an acid, and that will make the red indicator turn yellow. There's also a lot of salt. It's a salt agar. So if the specimen is halotolerant or halophilic, it can grow on the salt agar. So the plate is selective for things that are halotolerant or halophilic, and it's differential because if it changes color to yellow, it means it can use the mannitol. And staph, like salt, it's halotolerant, and it can use mannitol. So we're looking for something that will turn yellow and grow on this plate. All right, so let's say that we did the inoculation on the MSA plate, and then we incubated it, and we got this result here on the bottom right. So that would be a positive for growth on salt and a positive for fermentation of mannitol. Then we would do the gram stain. So let's say we did the gram stain, and we looked at the smear, the stained smear under the microscope, and we see clusters of gram-positive cocci. That would be a typical expected result for staph. All right, the catalase test. The catalase test is new. you take a little smear, a little loop full of growth off of one of your plates, and you smear it onto just a plain glass slide, like a microscope slide. And then you just drop some hydrogen peroxide on top of it. Some bacteria produce an enzyme that can turn the hydrogen peroxide into oxygen gas. And so if that enzyme is there, it will bubble. So this picture shows it bubbling. And so that would be a positive for catalase. The name of the enzyme that can convert hydrogen peroxide into oxygen gas is called catalase. It goes by another name, peroxidase, which may be familiar to some of you in if you took 1406 biology here at Richland. But if it bubbles, that means it does make that enzyme. So this example would be a positive for the catalase test. Then you have some antibiotics and some blood agar. You guys are familiar with antibiotics. So the Novobiocin would be an antibiotic that we would use to test the specimen. And you'll remember that you smear the specimen all over the plate, and then you drop the antibiotic disc on top of that plate, and then you let it grow and you look and see if there is a zone of inhibition. And usually you have to measure a fairly large zone of inhibition in order to conclude that the specimen is sensitive to that antibiotic. So that's an antibiotic test. In this case, the name of the antibiotic is Novobiocin. We usually look for about a 17 millimeter zone of inhibition in order for the staph to be considered sensitive. For this, the Columbia nalidixic agar plate, that's a plate that contains blood, whole blood, so it's going to be red, and it has this antimicrobial chemical called nalidixic acid. And what this is it prevents the enteric bacteria from being able to grow. So those gram-negative bacteria cannot grow on this. So the gram-positives, such as staph, should be able to grow on this plate. And it has blood, and staph typically will fall into one of two categories. Staph are usually either beta hemolytic or gamma hemolytic. Beta means they clear, they lyse all those red blood cells that are in the blood agar, and they create a clearing spot. Or gamma hemolytic means they don't. There is one other kind of hemolysis called alpha that turns kind of green, but staph is usually not alpha hemolytic. So we're looking for a beta clearing or no hemolysis. So let's say that you spread your sample on a CNA plate, and let's just look at the hemolysis. So for example, all of this that's clearing, it has this little clear zone around it, that's beta. Now this guy over here, that could be a gamma because it's not doing anything. So this is a good example of mostly a beta hemolytic. But remember, since you're taking a swab from your nose, you might get a couple different things. So it wouldn't be unusual to get a mixed culture. But this one is mostly has this, this beta hemolytic. The other thing they did here was they put the Novobiocin. She put it on there as conceptually, pretend like it was there. And she put the Novobiocin on, and she found a 25 millimeter, let's say, zone of inhibition, and that would be, it would mean that the specimen is sensitive to the Novobiocin. Usually we look for 17 millimeters or greater to determine sensitivity. All right, so did we check all of these things? Is your specimen gram-positive? Yes. Cocci, clusters? Yes. Catalase positive? Yes. Does it grow in salt? Yes. Is it either beta or gamma hemolytic? Yes. So these are the typical things for staph. And so once you confirm that you have staph, then you want to narrow it down to what species, because staph is just the genus. So let's narrow it down. The species that we're going to distinguish between Staph aureus, Staph epidermidis, and Staph saprophyticus. And so when you look at these, there's a few more tests. We already saw beta hemolysis. If it was Staph epidermidis, it would be a gamma, and Staph saprophyticus is a gamma hemolysis. I don't know why this is blank, but they can all grow on salt, so these should be plus, plus, plus. Do they turn yellow on the Mannitol Agar? Yes. This one is a yes. Epidermidis is a no. And then there's some other tests. So sensitivity to Novobiocin, we already did that test. Sensitive, sensitive. So there's a couple other tests we would need, typically. And the one's called coagulase, one's called SM110, and one's called DNase. So we're going to look at those a little more closely. SM110, SM actually stands for Staph media, recipe number 110, I guess. So Staph media 110. But what it is it's a particular recipe of agar, and staph grows on it. It does have salt, so it's a salt agar again, but it has something in it, and I can't remember what it is. If it is Staph aureus, it will pigment the colonies, and it will turn a golden yellow or an orangish golden yellow. And so let's say that you inoculated just a straight line, just a straight line down this plate, and that's usually how we do these plates, just a straight line. And we grew it after 24 hours, we took it out, and you see this golden yellow or orangish. That would be positive for the pigmentation. If it's a non-pathogenic staph, it will just be white like a normal bacteria color. But pathogenic staph tends to, like Staph aureus, tends to turn this golden yellow or orange. All right, what about this? DNase. DNase is short for deoxyribonuclease. That's an exoenzyme, an enzyme that the cell secretes, and it breaks down. So the plate has DNA in it, and the DNA is mixed with a stain called methyl blue or methyl green, but I think this picture is actually a methyl blue. But in any case, if the cells are secreting, you can see the area of growth, this little zigzag and then this straight line on this plate. If you can see right around it, the blue has sort of been broken down. It indicates that the DNA has been broken down, and you get this clearing. So this picture, this is a positive on the DNase test. If you see the blue color is uniform all over the plate, even where the growth is, that would be a negative. But a clearing means that these cells have secreted something, in this case, DNase. The last test is called coagulase. It's kind of a weird test. You take a loopful of your specimen, you put it into rabbit plasma. You probably know that plasma is the liquid in the blood when you remove the cells from the blood. And so you take rabbit plasma. I don't know who first figured this out. But if you take rabbit plasma and you put a little loopful of bacteria in there and you let it go for 24 hours at 37 degrees, if it is a Staph aureus, it will turn that plasma into sort of a gel. It will coagulate or kind of turn it into kind of a semi-solid blob. So in this picture, the uninoculated is all liquid. Then you put the loopful in there. If after 24 hours it's still liquid, then that's a negative. But if it turns into this blob and you have to kind of tip the test tube over to see it, then that would be a positive for the coagulase test. So it's not a color change in that case. You're looking for that liquid to turn into sort of a semi-solid blob. Okay, so on this example, we had beta hemolysis. We had the Mannitol Salt Agar turned yellow, so that's a positive. We saw a coagulase reaction. We saw golden pigment on the SM110 agar. We saw a DNase positive, and we saw sensitivity to Novobiocin. So all of those things indicate that this was Staph aureus. Now, on unknown 2 and unknown 3, you're going to go through the same process and see what species, and in this case it's going to be one of these two each time, and see if you can narrow down, but the purpose is for the practice.